What I want to try and do is give a fairly high-level overview both of kind of where genomics is at as a field and some sense of the research in the field over the last 10 or 20 years, which have got us to the stage where there's, I think, an enormous amount of potential for genomics impacting on clinical medicine. I'm aware that there's a range of different backgrounds of people in the audience, and I'll try and aim at a fairly general level, so that for those of you who know lots of the science already, please just bear with me, and apologies for repeating things that you're already familiar with. So I think it's helpful in terms of genetics and human disease to think of a spectrum, where at one end of the spectrum are diseases where genetics is the whole story. If you inherit one, or in some cases two copies of a mutation, a changed version of the gene in question, then you definitely get sick, and examples of those that we know of are cystic fibrosis, Huntington's disease. So those conditions are typically severe and individually relatively rare. Not so true for cystic fibrosis, but certainly for the other so-called Mendelian diseases. And though they're individually rare, collectively they account for a non-trivial proportion of the healthcare challenge that we face as a population. There's an estimate that 1% of people born will suffer severely from some kind of inherited condition. In many cases, we now know which genetic variants are involved. So the first success story there was the identification of the gene involved in cystic fibrosis in 1989. And it's a really salutary lesson that although the gene was identified in 1989, if I was giving this talk a year or two ago, I would have been saying there are yet no good treatments for cystic fibrosis. It's nonetheless been very helpful to identify patients earlier than would otherwise have been the case because of the genetics. Actually, last year, I think, the first treatment for a subset of cystic fibrosis patients who have a particular mutation, not the most common one in Caucasian populations, was announced, and it's had very dramatic effects on those with the disease. And there are a second set of treatments for others that are just being going through the regulatory processes in the US, so I think probably by this summer there'll be another drug for a much larger proportion of cystic fibrosis patients. So that was from 1989 until now, 25 years or so, to go from the genetic identification of the gene to actually using that in improving outcomes. Then in the mid-90s, with the discovery of two genes that you'll be familiar with, associated with inherited, high inherited risk of breast cancer, BRCA1 and BRCA2. And as you're probably aware, individuals who carry damaging mutations in BRCA1 have a much increased lifetime risk of breast cancer, up to about 80%, and a very high risk of ovarian cancer as well. And again, the identification of the genes, because of those large effects, the identification of the genes had an immediate clinical impact for different healthcare systems handle this in different ways, but for individuals at high risk where they choose to do so, they can be tested. Some of the individuals, even if the mutation is in their family, will not carry the mutation. That'll be a 50-50 event. Those that do can then use that information in conjunction with their healthcare providers to work out what steps they'd like to take to ameliorate risk. And the whole issue is achieved in a very good way, achieved much more publicity recently through Angelina Jolie, telling people very directly of her experience in learning about a BRCA mutation. You'll have heard, I'm sure, of the Human Genome Project. Many of you will know some of the details. So the Human Genome Project aimed to read one copy of our genome, of the entire set of DNA that we carry. Our genetic code, as many of you will know, is broken up into nucleotides, so individual letters in a DNA alphabet, and we inherit 3 billion of those from our mother and 3 billion from our father. You probably also know, so we'd like to think of ourselves as amongst the more complex creatures on the planet, and we probably think that needs a lot of DNA to encode that complexity. Rather humble salamanders have much more DNA than we do, as do tomatoes. So there's not a direct correlation, or salamanders, if when they're talking to their colleagues may take a slightly different view, there's not a direct correlation between the amount of DNA we carry and the complexity of the organism. So the Human Genome Project read, as it were, the DNA that we share, what we have in common. If you compared one of your chromosomes with the chromosome of a person sitting next to you, or indeed one of your chromosomes with the other copy you have of that chromosome, the DNA sequences would be identical at about 999 positions out of 1,000. So there's a lot of similarity, but that's still lots of differences, and it's those differences that are responsible for inherited differences between us, including differences in the way we look, certainly some of the differences in our susceptibility to disease, and potentially behavioral traits as well. And a lot of the focus in the field over the last 10 or so years has been in understanding the relationship between that variation and health outcomes and disease susceptibility. So I said at one end of the spectrum are the so-called Mendelian conditions, where genetics is all the story. If you inherit the wrong version of the gene, you get sick. At the other end of a spectrum are all of the common diseases in humans: heart disease, diabetes, arthritis, schizophrenia, and so on, many of the cancers, where genetics is part of the story about why people get sick. We've known that for a long time because those diseases run in families more than we'd expect by chance. And recently there's been a large effort to try and understand exactly which letters in our DNA make a particular individual more likely to get heart disease and someone else more likely to get diabetes or schizophrenia. So although there was a lot of research after the BRCA discoveries, for example, there wasn't much progress until about 10 years ago now. So I'll walk you through a bunch of slides like this. So these are ideograms of the human chromosomes. And before 2005, there were maybe 5 or 10 or 15 examples of specific genetic variants, letters in the DNA, that we knew were associated with changing people's risk of inheriting a disease. And then in 2005, in what we think of as a kind of new age, because we were able to do experiments in a new way, that picture changed. So I hope you can see a little lollipop stuck on chromosome 1 here. That indicates a finding in 2005 of a change in DNA letter at that position, which affected susceptibility to a particular disease, in this case, a disease called age-related macular degeneration. That was 2005. There are a few more examples in 2006. The picture changed quite a lot in 2007. That was 2009, 10, 11. And 18 months or so ago. So we went from knowing 5 or 10 examples of changes in DNA associated with disease susceptibility to now a catalogue of over 4,000 variants associated with literally hundreds of different diseases. So we found the DNA variants involved. They, as I'll come back to say, they affect disease risk, but usually, and for most of these, not in a major way. There's a list which you won't be able to read of all of the conditions involved, and as I said, there are hundreds of those. Everything from the major conditions like heart disease, diabetes, the mental health disorders, arthritis, autoimmune disorders, and many of the common cancers, and then also conditions to do, or traits like height and weight and so forth. And you'll notice that there was a kind of acceleration in those findings in 2007. Every year, Science magazine, Science and Nature are the kind of two main general magazines in our field, and the ones that as academics we aspire to publish in. Rather pompously, Science magazine each year decides at the end of the year what was the most exciting breakthrough in all of science for that year. And they pick this explosion, and now we see it as the tip of the iceberg in our understanding of the genetic basis of common disease as the major breakthrough in 2007. And to give you a sense of what changed there, we were able to do large studies where we think several thousand individuals with a disease in question, maybe diabetes, several thousand individuals either explicitly without the disease or sampled from the population. We measured them at many places, 500,000 or 1 million places in their genome, and we just looked for DNA letters that differed in frequency between the sick people and the healthy people. And the rationale is kind of not rocket science. Well, it's not rocket science in the obvious sense, it's biology rather than astrophysics. It's also not rocket science in the colloquial sense. If there is a DNA variant that makes you more likely to get a particular disease, then that variant should be more common in people with the disease than in healthy people. And that's the difference we were able to look at. And the big change was our ability around that time, 2005 or 6, to measure large numbers, 500,000 or 1 million positions in the genome, relatively cheaply. Why do we care? Well, I think most of us would say there are two different sorts of reasons. Let me take the second one first. As I said, these genetic variants affect risk of disease, so you might think that it would be quite helpful to measure them in healthy people and predict which diseases they'd be at higher risk for and lower risk. That's possible, but currently most of the genetic variants we know and have identified have small effects on risk. They might increase your risk of, say, heart disease by 5%, or someone else's risk of schizophrenia by 3%, and so on. So individually they don't have a big effect, and even collectively often they don't change risk by large amounts. So it's very different from the Mendelian world, where if you get the mutation you'll be sick, or even the BRCA mutations, which have a big impact on risk. So by and large, these variants from a health point of view haven't been particularly helpful for predicting disease risk. That will change over time as we learn more about our DNA and the way that's related to disease risk. It's still the case for most diseases that knowing family history is a better predictor of disease risk than knowing these particular variants. On the other hand, and for most of us in the field this is the more important of the two reasons currently, on the other hand, each one of these is a clue to human biology. If we find out that having a T rather than an A in this position makes someone more likely to get heart disease, we can look at what's going on biologically in individuals carrying the T there rather than the A, and we will often get completely new clues and insights into the pathophysiology, the path to heart disease. And that can point to new treatments. And in fact, one of the really interesting things about the many findings that we've had, although in very few of them do we fully understand that biological pathway from DNA to disease, in many of the instances, the genes that are involved just weren't on people's radar as being important for the diseases in question. So there's a huge amount we have learned and much more for us to learn that in time I think will lead to new drug therapies. I said the studies we've done up till now have typically looked at a few thousand individuals with one disease and compared them to healthy people. That's changing. It's changing both in terms of scale and what's measured on people. So we're now moving into a world, and I'll give you a couple of examples in a minute, where it's possible to measure many, many different things on individuals, either by explicitly measuring them or by just using the individual's electronic medical records and comparing that with DNA that we can measure on the people. So around the world, there's a large project in the UK Biobank, which some of you may be aware of or potentially even participants in, that's taking 500,000 people in the UK, aged between 40 and 70 at recruitment, measuring lots of information on them at baseline. We're in the process of completing the measurement of extensive genetic information on those individuals. There'll soon be extensive imaging. So a really powerful resource to understand not just the genetic basis of disease, but the way that interacts with lifestyle choices and so on. And there are other projects in other parts of the world, in the US, in China, a large biobank in which our colleagues in Oxford are very centrally involved, in Japan, in Mexico, again in the Mendelian Veterans Program in the US. In the UK, and I'll come back to this, there's also a project to sequence the entire genomes of 100,000 patients in the National Health Service. As I said, we're not just collecting disease information on these individuals or genetic information, but extensive and quite detailed information about metabolites, about cancers, and even medical images. So what's been driving this? There have been changes in technology, there have been changes in our ability to analyze these large amounts of data, and those challenges are getting bigger, and changes in our biological understanding. So there's a picture that's often shown that gives a sense of the cost of DNA sequencing. The Human Genome Project read one version of the human genome. It was completed in 2003. It cost something like $2 or $3 billion and took hundreds of scientists from many countries about 10 years. So it's now possible to sequence a human genome effectively overnight, and the cost is something like the cost of getting the data, not of analyzing it, is something like £1,000. So there's been an extraordinary change, which this indicates, and that's what's allowed genetics and genomics, first of all, to power the large research studies that have been going on, and then to move on to look at the possibility of measuring genetic information extensively in the context of clinical medicine. So personalized or precision medicine is a common theme. Some of you will have been involved in a conference that's just finished here on that context. I just want to give a couple of examples. So one of them has to do with our genetic makeup and how that relates to the way we react to drug therapies, either in terms of how effective a drug therapy is, or potentially whether we suffer from adverse side effects. Another key area where genetics is having a major impact is in cancer. As you'll be aware, cancer is all about the genome. So what happens in cancer cells is that their genome is messed up. They've got an instruction manual different from the cells, the normal cells in that person's body, and they now differ in ways that we can measure. So it's possible to read the genome in someone's cancer and to compare it with the genome in their ordinary cells and look at the differences. So here's one picture. So genomes are big, 3 billion letters. So what I'd like to do is show you the 3 billion letters in someone's cancer and the 3 billion in their ordinary cells. So here's a way of trying to picture that visually. So here the genome is represented around the outside of a circle. There are marks on the inside which show large chunks of chromosomes in the tumor cells which aren't where they should be. So a bit of chromosome 6, instead of being joined to the rest of chromosome 6, is stuck on the end of chromosome 18, for example. Then there are kind of medium-sized things which are millions of DNA letters long, which are duplicated or deleted from the genome in the cancer cells. And finally, there are about 30,000 changes in single letters around the genome. So the high-level point is very, very gross changes and differences between the cancer genome and the ordinary genome. And in fact, this picture, the last picture was for a particular melanoma tumor. These are the changes in the genomes of breast cancer tumors in 6 different women. And the high-level point I want you to observe is they're very different. The sorts of changes that go on are quite different. So we're moving to a world where we can think of cancer very much in a personalized way. We can understand what are the changes in a particular individual's genome and cancer genome, and try and target therapy at that, rather than, as has been the case up till now, classifying cancer by where in the body it occurs. And indeed, it may be the case at the genomic level that one person's breast cancer has got more in common with someone else's lung cancer than with another breast cancer patient. There's one of the most interesting examples, although there's a caveat, is this one, which I hope you can see part of. So many melanoma tumors have a change of a particular type. So this is a kind of genetic language that says in this gene BRAF, at a particular position in the protein, one amino acid is changed to another one. And that effectively leaves the gene turned on and causes proliferation of the tumor. And the picture, if you can see it, is a scan of an individual with advanced melanoma that's metastatic with tumors effectively everywhere. So the discovery of this genetic change in tumors happened in about 2001 or 2002. And then by about 10 years later, people had developed a drug, a so-called BRAF inhibitor. So what's going on in the tumor is too much of this protein BRAF is being produced. So drugs introduced that kind of mops that up, as it were. And when it was given to patients in a clinical trial, so here's someone with advanced metastatic cancer. Here's the same individual about 2 weeks later after this BRAF inhibitor. And the change is extraordinary in that most of the cancer has gone away. So that was exciting and encouraging at the time. What we now know is that, and kind of suspected at the time, cancer is quite clever. And if you looked at a similar scan from this or another patient 6 months or 9 months later, much of the cancer has come back because the cancer evolves itself a way around the drug and develops resistance to it. So it's on the one hand encouraging in providing a potential therapy for melanoma. It's now used, I think, in combination with others in helpful ways. So I just want to conclude by talking about a big initiative in the UK, and I'll put it in the context of a study we did in Oxford now 4 years ago with Illumina, the big sequencing company, to try and understand what were the near-term possibilities for reading entire DNA sequences in clinical medicine. So we kind of did an experiment of saying to the doctors in the hospital in Oxford, so if you think you've got a patient where genetics might be a part of the story and you can't work out what's going on currently, we'd offer to sequence the genome of that patient and where relevant and of relatives to try and work out what the particular changes might be relevant to either diagnosis or potentially treatment. We did that for rare diseases and for cancer. So here's one early example of a young girl who had extensive problems through craniosynostosis. We sequenced both of her parents and the young girl herself, and we found what's called a de novo mutation, so a genetic change in her genome that resulted from an error in copying either the father's genome or the mother's genome and passing it on to her. In this instance, it didn't help treatment. It was helpful for the parents to know what had caused the problem, that it wasn't something they'd done during pregnancy or at some stage early in their daughter's life, and also very helpful in terms of counselling them for risk of future children. So because this was a kind of one-off event, the doctors were able to tell the parents that they wouldn't be at increased risk for subsequent children, which is obviously very helpful for them. So that attracted some attention at the time as an early example of genomic medicine in the clinic. And I'll just conclude by saying it's led, as have other efforts like this in other parts of the world and in the UK, to a very large project in the UK where the National Health Service is now sequencing the genomes of 100,000 people, focusing initially on cancer and rare diseases, an area I haven't talked much about but is also interesting, on the sequencing of the genomes of bugs, the bacteria that make people sick. Thanks very much.